So, anybody have any questions? Did everyone get my email? No, the one I sent out this summer.
I was like, wait.
I didn't send it. Any questions about syllabus? Okay, don't forget to sign your disclaimer form at some point and get that to me. You should consider these printed lecture notes that are posted online part of your lecture notes. And since we don't have a book, I will consider this fair game for exam material. Most of it, if not all of it, will be covered as we do our lecture section, sometimes not always in the same order. But I would encourage you to read these prior to when we start a new section, and then that way you can kind of be on the lookout for, did we cover this, did we cover this? And then if there are any questions, because these are a little sketchy, they're not written out in great prose, and so if there's something that doesn't make sense in these, if there's something that we maybe didn't cover and you're not quite sure about, then I can fill in some gaps for you. But I might not in class cover every absolute thing in here, but I consider this to be pretty essential information, okay? And probably beginning about next week, I'll start making some more intentional recommendations about things you should be looking at on mastering microbiology to kind of supplement this, okay? And that would be both for lecture and for lab. Wednesday, I would like you to come to lab. It's going to be a very short lab period because a bunch of us are going to the funeral, and it's at 10 o'clock in Shelbyville. But what I'd like to do is give you just a little brief introduction, and then I'm going to have you, while I'm gone, restock your lab drawers and make sure you've got everything that you need. So I'll give you some sheets of paper and kind of show you where things are right when lab starts, and then when you get that done, I'll have you pick out a microscope and label that. And so there'll be a few things to do on Wednesday, but it won't take the full 2-hour period. And then Friday, we'll just have a lecture because we don't have a lab scheduled. Labs will begin next week. Okay? Nothing? Okay, let's continue where we were last time, spending a little bit more time on the why question of why bother studying microbiology and what are the relevance and importance of microorganisms. I think that if you ask the general public and even a lot of science students what's important about microbiology, the big thing that they would center on immediately and maybe almost exclusively would be that they're potential pathogens. Okay? Are all microorganisms pathogens? No, actually a small tiny fraction of them are pathogens, but that's what we tend to concentrate on. And we'll talk about that a little bit more in a minute, and that will be a theme throughout the course, but I want to concentrate a little bit more now on some of the other roles that microorganisms play. And we started, excuse me, we started to flesh that out a little bit last time, and I would like to continue that today. It only takes me about a week to get my voice back after the summer. So, one of the things we were talking about is the potential ecological significance of microorganisms. And we had a couple of columns going where one was positive benefits, one was negative benefits. And we were talking about decomposition, negative benefit associated with food spoilage, crops being lost in the field before they even get to market. But on the other hand, nutrient recycling is an absolutely essential aspect of all life on Earth. There are a set of very complex chemical and physical properties working in our environment called biogeochemical cycles. And that's a fancy word basically for nutrient recycling. So there's a carbon cycle, there's a nitrogen cycle, there's a phosphorus cycle, there's a sulfur cycle. Okay? And all of these cycles involve nutrients moving through various compartments of our worldwide ecosystem, oftentimes from soil to living organisms back to soil again. But air is involved, obviously, in the carbon cycle. And so these cycles are just essential for life on Earth. And microorganisms play a really critical part in these biogeochemical cycles. That is particularly related to microbial metabolism. And so when we talk about microbial metabolism later, we're going to come back and kind of superimpose some of these cycles onto metabolism and see the important role that microorganisms play. One of the ones we're really going to concentrate on most heavily is the nitrogen cycle. And we'll talk about some phenomena like nitrogen fixation and deamination and ammonification and all kinds of different bacteria that are involved in this complex cycle that moves nitrogen throughout various aspects of our ecosystem, particularly from the soil to living organisms to the atmosphere and back again. And so we're going to try to really make microbial metabolism relevant to some real world type of applications. I mentioned photosynthesis as being an essential ecological process. And we will spend some time talking about bacterial photosynthesis. Some bacteria carry out the same type of photosynthesis that higher plants do, but most bacteria carry out a different type of photosynthesis, which has some real wrinkles. And so we're going to talk about some of those differences between quote-unquote oxygenic photosynthesis and anoxygenic photosynthesis. A lot of bacteria photosynthesize but don't produce oxygen. Fortunately, there are a bunch of bacteria and other single-celled organisms that are oxygenic photosynthetic bacteria, and as I said at the end of the period, they produce about 50% of the oxygen on the planet. And oxygen obviously is important not only for cellular respiration, but what other essential ecological role does oxygen play? Pardon me?
Atmosphere.
Has to do with the atmosphere. Absolutely. Under the influence of especially ultraviolet radiation in the atmosphere, oxygen atoms can interact with one another to form a molecule O3. What is O3? Ozone. Okay. And ozone we sometimes tend to think of as being a pollutant, and it can be in the lower atmosphere. Ozone can be produced by combustion. LA was famous for ozone at one time. Produces this kind of gaseous brown smog that LA was famous for. Getting a little bit better. But in the upper atmosphere, what role does ozone play?
Does it reflect ultraviolet light?
Yep. Ozone filters out various forms of harmful radiation, including ultraviolet light. Until photosynthesis evolved, and until we had then an ozone shield, could life exist on terrestrial environments? Nope. So where did life have to live? Water, because water will filter out ultraviolet radiation as well. And so that evolution of photosynthesis, the development of the ozone layer, then resulted in an environmental modification that allowed life to then colonize land. And so obviously a very important ecological phenomenon. Another thing that's produced during microbial metabolism is gaseous methane. Methane will move up into the atmosphere. And methane, like carbon dioxide, has an important property in that it traps heat in the atmosphere. Why is that important to us? Global warming, global climate change, if we trap too much heat. But again, could life on Earth have evolved without methane and other gases like CO2, which are also produced by microbial metabolism? No. We need an atmosphere to retain the radiant heat that the planet absorbs. Otherwise, if you don't have an atmosphere, like Mars lost its atmosphere, all that radiant heat is just re-radiated back into space again. So we have this uninhabitable temperature extreme on Mars that we don't have on Earth because of the blanket of gases that trap radiant energy. We have a very critical balance that needs to be achieved between trapping enough energy to make the planet habitable, but not trapping too much energy to cause atmospheric warming. any relationship between atmospheric warming or global climate change and bacteria? Yeah, there are. Because now many of the hosts of some pathogens are changing their range because of climate change, and we're getting hosts that can transmit disease moving into new environments. And so global climate change has an effect on microbial populations. So there's a bunch of ecological relevance to microorganisms. There's also a significant amount of industrial relevance to microorganisms. Think of any examples? How are microorganisms used in industry? Austin?
Like a sewage treatment facility?
Absolutely. Okay, water treatment. And there is a whole complex area of microbiology that has to do with setting up systems which are tended to kind of mimic natural systems, where if you put organic matter like fecal material into bodies of water, that microorganisms through the metabolism will degrade those and recycle the nutrients. What happens when we start developing cities and large population centers is that if we just discharge the waste into bodies of water, we can quickly overwhelm the natural system, and therefore we've got to supplement it with sewage treatment facilities to augment that particular natural process. And so waste treatment and water microbiology is a huge applied area that we'll mention a little bit about in a couple of contexts. Any other industrial applications you can think of? Yes?
Like water.
Oh,
sugar,
yeah,
beverage.
food. And let's go ahead and put beverages in there as well. Microbial foods and beverages are primarily produced as a result of microbial fermentation. And what's probably the example that comes most to mind when you're talking about microbial beverages? Beer, wine, various forms of alcohol, yeast fermentation. Okay. What about microbial dairy foods? Anybody know what kind of microorganisms are involved in producing yogurt, cheese, cottage cheese, sour cream?
I know it's genetically modified.
They can be, yeah. They have been, some of those have been genetically modified. But they're not producing alcohols as a process of their fermentation. Instead, those organisms, like an organism called lactobacillus, which can ferment lactate, is producing acid compounds. And when lactose is fermented in milk, for example, then the fermentation of the lactose produces acidic compounds, which causes the milk proteins to precipitate. And depending upon how that happens and the conditions under which it happens, we can form different products like cheeses or sour cream or etc. Okay? So a lot of foods are produced as a result of microbial metabolism. And there's a couple of more examples in the notes that I've written out for you. Various chemicals can be produced which have industrial application. Microorganisms as a whole can produce a really impressive array of chemicals as a result of fermentation. Okay? When we talk about fermentation with eukaryotes, we primarily think about either ethanol fermentation or lactic acid fermentation. But microorganisms produce all kinds of weird and wonderful things as a result of their fermentative processes. They can produce alcohols, they can produce acids, they can produce antibiotics, they can produce hormones, they can produce enzymes, which are used in a variety of industrial processes. And all of these, at one point in time, have been harnessed because it's fairly easy to grow up big vats of bacteria, and if you give them the right food and grow them under the right conditions, and you have the right strain of bacteria, then they can crank out large quantities of things like ethanol and acetic acid and butanol and you name it. There's a whole bunch of industrial products. Or various enzymes that are used in cheese production, or dyeing blue jeans, or you name it. Again, there's just all kinds of different applications industrial. And I think there's a couple of PowerPoint slides again here that show you. Yeah, these are some of the industrial products of microorganisms. There are some antibiotics that are listed right there. Some various enzymes that are produced, catalase, amylase, proteases, some various acidic compounds that are being produced. And so there's a whole just huge industrial complex of chemical production that is associated with microbial metabolism. And again, as we talk about metabolism, we'll flesh that out a little bit and talk about some of the more interesting applications. One little story that I think is quite interesting, and it's kind of outlined in your notes a little bit, is that in some respects, you can really kind of credit World War I, the Allies winning it, to a discovery in microbial metabolism. There was a guy who figured out how to produce acetone as a result of microbial fermentation. And acetone is used in making gunpowder. And gunpowder was really one of the limiting factors in World War I, and whoever could produce the most gunpowder had the most chance of winning the war, and that ended up being the Allies. World War II, a lot of World War II, particularly in the Pacific theater, really turned upon the ability to allow soldiers who had been wounded to survive battlefield infections. And that primarily was the result of research that had been done and then marketing that had been done in the production of antibiotics. And so that really changed the tide in World War II. And so these little tiny critters can really have a tremendous influence on historical events in human populations. The Black Plague. Okay. There are some other interesting applications that have to do with kind of business. Microorganisms are involved in mining and extracting ores. As they metabolize materials, they can turn them from oxidized to reduced states. And the oxidized state of metals tend to be more easily extracted than the more water-soluble reduced states. Also, after you mine ore, sometimes micrometabolism produces pollutants. There's a phenomenon called acid drainage, which is associated with coal production. And in coal production, some of the mine tailings that are produced and oftentimes not disposed of properly contain high sulfur coal that's not used and is kind of thrown away. And microorganisms can metabolize that sulfur in the coal and produce sulfuric acid. And the sulfuric acid can leach into waterways and cause all kinds of ecological problems. So we could spend all semester just talking really about kind of industrial microbial applications. And in larger schools, there are courses, particularly at the graduate level, on industrial microbiology. We've taken that a step further, too. Sometimes we're not satisfied with just using the wild type form of the organism to do something for us. We genetically modify it. Okay? That has led into the whole use of microorganisms in applied biotechnology and in molecular biology research. So that's another use of microorganisms. Remember battling through protein synthesis, DNA replication, all that stuff in cell biology? All that stuff was worked out initially using microbial models, particularly E. coli. E. coli is kind of considered to be the lab rat equivalent to a physiologist. Okay? We know more about E. coli than we do any other bacteria. Microbial cells have a pretty simple genetic system, and so if we can work out the steps of translation, transcription, DNA replication in that simpler organism, then it's easier to then apply what we've learned to the more complex eukaryotic genomes, and we have done that. Then in the 70s, we learned how to transfer genes from one organism to another via transformation. And you now do that in cell biology. You do your PGLO lab and your various exercises where you change the phenotype of an organism by inserting a gene into it. Eli Lilly made a whole boatload of money and is still making money on its Humulin, human insulin. By being able to isolate the human insulin gene, implant that into a bacterium, grow up vats of bacteria, and then isolate the secreted insulin from those particular vats of fermentation. What's the advantages of Humulin versus porcine or pig insulin? What's the risk you take if you inject some pig insulin into your circulation? Yeah, having an antibody response against that, and some people don't tolerate that pig insulin as well. The closer you can get for a species match, the better on. Some of you have taken Dr. Morton McComb's biotech class, and all of the biotechnology applications which go into medicine, agriculture, are just exploding. And again, without microorganisms, that would not be possible. So a lot of economic things associated with the importance of microorganisms. In 1964, the U.S. Surgeon General told Congress, it's time to close the book on infectious diseases. The war against pestilence is over. Did the Surgeon General get it right? Nope. Anybody have any insight into why a Surgeon General, who was a highly accomplished MD, could have gotten it so wrong, and what was really the basis for his optimism? Why did you think the war against infectious disease had been won? Oh,
Maybe just from how well they developed antibodies, they didn't think it was a lot of
antibiotics was a huge part of it, okay? Entomologists made a similar claim at about the same time with DDT. DDT was this miracle wonder drug, and when you sprayed it on insects, they just died immediately, and they really thought that they could pretty much eradicate insects by the use of DDT. The Surgeon General thought the same thing, because antibiotics were just this remarkable new tool for killing microorganisms. Okay? So, why did the Surgeon General get it wrong? What did he miss?
their ability to evolve.
The evolution of antibiotic resistance was a big part of it. Some people consider this to be one of the looming largest public health threats that we've ever faced. It's the evolution of antibiotic resistance. You've all heard of MRSA before, right? What does MRSA stand for?
Resistant Staphylococcus.
Methicillin-resistant Staphylococcus aureus. Okay. Methicillin is one of the penicillin antibiotics. Okay. Why do microorganisms develop resistance? And why should we have expected this to happen, just like we should have expected it to happen with DDT? Natural selection. Microorganisms evolve resistance. Natural selection, remember, has to have variation to act upon. There have to be naturally occurring resistant forms in a normal population, and then the presence or the application of that antibiotics favors them over the non-resistant forms. What's the source of variation, particularly in microorganisms? Where do those altered resistant forms come from? The antibiotic doesn't produce them, does it? It acts on ones that are already there. What's the source of those varied forms. What process produces them? Mutation. And mutation is naturally occurring. And in microorganisms, they tend to be a little bit more prone to show the effects of mutation immediately because of the nature of their genome. How does a prokaryotic cell's genome differ from ours? What's the nature of our genome? How many chromosomes do we have? How many chromosomes? How many human chromosomes? 46, 23 pairs, right? So we are diploid. And so if you have a mutation in a gene, is it necessarily then expressed as a change in phenotype? No, it might be a recessive mutation, right? How many chromosomes does a typical prokaryotic cell have? One. It's usually a circular single chromosome. They have a haploid genome. And so if a mutation occurs in those genes, is there any other gene to dominate? No. And so mutations oftentimes really show up much more readily in prokaryotic cells than they do in eukaryotic cells. And because those cells reproduce so rapidly, remember generation time about 20 minutes? These microorganisms can respond to selective pressures and evolve very, very quickly. So the evolution of antibiotic resistance has been huge. There are some other things that the Surgeon General missed as well. We're living longer. And the longer you live, the more likely it is that you're going to contract disease, particularly cancer at some point in time. How come? How is age related to susceptibility to disease? Cells are dividing and prone to mutation, but just talking about maybe infectious disease or cancer.
Longer exposure.
Longer exposure potentially to potential pathogens, greater just statistical chance of being exposed. That's a good possibility. Oh. Your immune system declines. Okay. My immune system is far less robust than yours is. Okay. When you're young, your immune system is developing. By the time you get to puberty and a little bit beyond, your immune system really is at its prime. And then throughout your life, it starts to decline. And so older people are more prone to infectious diseases and developing cancer. And we'll talk about why that's the case when we talk about the immune system at the end of the semester. There are some interesting things going on with diseases called emerging or re-emerging diseases. Emerging diseases are kind of new diseases that pop up, sometimes almost overnight. Viral diseases are sometimes emerging diseases. When I was your age, I'd never heard of Ebola. Okay, we still don't know really where Ebola came from and why Ebola seems to kind of go dormant for a while and then pop back up again. In the 90s, there was a disease outbreak in the southwest United States called hantavirus. That was a new disease. And sometimes diseases that we think we have licked and beaten into submission come back again. And that is the case particularly with tuberculosis. Tuberculosis in the 20s, 30s, and 40s was a scourge. And we had sanatoriums where people were put to treat and isolate them from other people if they had tuberculosis. What changed the tide in tuberculosis? Why did sanatoriums disappear? Why did tuberculosis almost disappear as a disease? Antibiotics. Okay? But resistance is developing. And so this is one of the re-emerging diseases that is becoming an increasingly large public health problem. And then there are some really what I like to call surprising connections. And that is, there are some human diseases that we didn't used to think were related to microorganisms at all. A great story is stomach ulcers. We used to think that stomach ulcers were caused primarily by stress and diet. If you eat too many spicy foods, you could get an ulcer. And so treatment obviously many times involved changing your diet, consuming things that might tend to be soothing. The idea for a while was if you drink milk, it would coat your stomach and help you to feel better. In the, when was it, probably 90s, a couple of Australian researchers developed some hypothesis that bacteria that could live in the lining of your stomach and tolerate the acidic conditions of your stomach were responsible for most stomach ulcers. And people just ridiculed them and said, microorganisms can't live there. That's impossible. And they did a lot of epidemiological research, but kind of the topper to their particular story was they did probably the most stupid thing a researcher could do. They were both healthy, didn't have ulcers. One of them chugged a culture of Heliobacter pylori, which was the suspected causation of stomach ulcers, and they got an ulcer. So now we have a new avenue for potentially treating ulcers, antibiotics. Okay. Various forms of heart disease are now thought to have a microbial implication, particularly if an infection causes inflammation. We're going to talk about inflammation a fair amount later in the semester. Some of you have encountered inflammation as a result of mechanical injury like a twisted ankle. Okay? It has those characteristic signs of redness, heat, swelling, pain. Okay? Everybody suffered from inflammation. But we think now a lot of diseases that we didn't think had a microbial component are the result of infection, and that results in chronic states of inflammation in your body. One of the things that they're recommending now is that people who have history of coronary artery disease in their family get an additional blood test, which they think might be much more predictive of coronary artery disease than cholesterol screening. And that is a test for something called C-reactive protein. And C-reactive protein is a measure of inflammation. So these protein levels rise when you have inflammation. So a lot of these medical connections that we hadn't made before are now becoming more and more fleshed out, and we understand the connection between new diseases and microbial components. So, unfortunately, the Surgeon General got it wrong. And then, so there's a nice necrotizing fasciitis caused by group A streptococcus bacteria, quote-unquote flesh-eating bacteria. That is one of the diseases that emerged about a decade ago. This shows you top causes of death for all diseases in the United States and worldwide. And you can see that some of these are clearly associated with microbial pathogens. Others are diseases of, we call them affluents. They might be the result of bad diet, exposure to toxins and things like that. But one of the things that you will see if you compare the two columns with one another is that worldwide there's kind of a different profile, and there's much more of a component of microbial related diseases than there is in the United States. We have a better public health care system. We have access to more drugs to treat these microbial diseases, vaccinations, antibiotics, etc. This again shows you a profile of developed countries versus developing countries. Look at the difference in the effect of infectious and parasitic diseases in developing countries versus developed countries. Okay? Whereas in the developed countries, diseases of the circulatory system, which oftentimes are diseases of affluence, bad diet, etc., are the most predominant form of disease. And so there's this whole relationship between disease and poverty, and access to clean food, clean water, the types of drugs. And Jake, you probably got some interesting insights into that with your experiences this summer, didn't you?
Yeah.
So anybody who has traveled overseas to less developed countries understands what this is all about and how important and what a role poverty plays in the process. Okay. Okay, well, like I said, we could spend all semester talking about relevance of microorganisms and why study them, but this gives you a little bit of insight into how pervasive the influence of microorganisms are on the world and our society. Let's briefly turn to the question of where. Where do microorganisms live? Standard pat answer is everywhere. Are there microorganisms on the outside of this Petri plate? Absolutely. On this paper, on my hand, in the air. Yep. The human body is a great place for microorganisms to grow because we are an incubator. We have a set of homeostatic mechanisms to maintain an internal body temperature of approximately 37 degrees Celsius, which is what one of our incubators is set at that we'll be growing unknowns at. We also have homeostatic mechanisms to control oxygen concentration to our tissues, maintain pH levels, maintain solute concentrations at set levels. And so we're a great incubator. There are some pretty extreme environments around the world, and I mentioned a couple of those the other day, where microorganisms could grow that you really wouldn't expect they could grow. Here are some microorganisms growing in ice sheets in Antarctica. They can grow in acids. They can grow in distilled water. They can grow in the upper atmosphere. For a long time in the history of the Earth, microorganisms were the only game in town. Okay? We think that the origin of the Earth was about 15 billion years ago, and that prokaryotes appeared on the planet about 3.5 billion years ago. Eukaryotes appeared about 1.75 billion years ago. So for approximately 2 billion years, prokaryotes had the world to themselves. They reproduced rapidly. They were able to adapt and fill virtually any niche on the planet that was available. They have remarkable adaptive abilities, and they have exploited those abilities to colonize virtually every habitat that you can possibly think or conceive of. Microorganisms sometimes get a bad rap. We think that prokaryotes are these simple organisms. And from a cell structure point of view, they are fairly simple. They're not as complex as eukaryotic cells. They don't have all those neat membrane-bound organelles. They don't have a mitochondria. So structurally, they're pretty simple. Metabolically, they put us to shame. Okay? Microorganisms have a much more sophisticated set of metabolic pathways than eukaryotic cells do. Because a lot of eukaryotic cells have become codependent on other cells and multicellular organisms, they have kind of given up some of those abilities. we don't produce a lot of things that microorganisms do, we consume them instead. But microorganisms can grow just fine, at least most can, on the simple growth media like agar or this tryptic soy broth, where basically they are supplied with a source of carbon and nitrogen, and they can make any molecule they need to make new cells and grow and reproduce. I'm sorry, but if I locked you in a room and gave you all the agar that you could eat, you would die. Okay? You do not have that metabolic capability of taking those simple compounds in agar and converting them into all the things your body needs to sustain itself. So keep that in mind. It may be structurally simple, but very metabolically complex. Well, the fact that microorganisms can live anywhere creates some challenges for us working in the lab, and I mentioned that yesterday. And so one of the early things we're going to do in lab is practice some very fundamental aseptic or sterile technique so that when you're working with your unknown, you don't contaminate. Microorganisms can live in the upper atmosphere. And one of the big questions that NASA is investigating with its various probes and landers to Mars is, could there be microbial life on other planets? And there's a lot of interest in some of the new exoplanets, the Earth-like planets that they've been recently discovering, and some of the moons of Saturn and Jupiter, where they found ice and water, and at least for life as we know it, you can't have life without water. And so there's some very, very high levels of excitement about the potential for discovering microbial life on other planets as we do more and more investigation. There's also an emerging theory that life on Earth may have been seeded by microbial life transmitted by meteorites, asteroids, comet material. And several years ago, there was a lot of controversy about this particular slide. A meteorite was discovered, and this image was taken of it, and a lot of people believe that this represented a fossilized cell, and that they saw a lot of those in these particular meteorites. There were other people that believed that these were artifacts of physical processes, and the jury is still out on that. But this whole idea of the potential for microbial life on other planets is really exciting, and I think we're going to find it. I mean, I think that if you have conditions of water and suitable temperatures, that microorganisms are so adaptive that it's very likely that they would have been present if they're not present right now. And that would be huge. So let's talk a little bit about the who and when of microbiology and do a little bit of background history. And I'm going to do this in such a way that's going to lead us into some applied discussions of how the various techniques that are fundamental to the development of microbiology arose over a period of time. When you talk about history, you really have to talk about the history of the development of microscope. Now, we don't know exactly who developed the first microscope. But there is an individual who is given a lot of credit for not only developing some of the first primitive microscopes, but then detecting microorganisms with that instrument. Anybody know what I'm talking about? The person who's kind of credited with being the discovery of the microscope. Anton von Leeuwenhoek. He lived from 1632 to 1723. So pre-Revolutionary War. He was a Renaissance man. He had a lot of interests. His profession was that he was a Dutch linen merchant. He sold cloth, linen cloth. And he developed his first simple handheld microscopes to inspect linen for small flaws. Okay, you ever seen jewelers with their little eyepiece thing to look for flaws and gems? Well, he developed a similar type of device for looking at linen. And with this single lens, he could magnify up to about 300x. And this is a model of the first von Leeuwenhoek microscope. You can see down here that it was just held up to the eye. And it was really basically a fairly sophisticated magnifying glass. And it had some little devices to hold specimens and a focus screw where you could change the focus level. Von Leeuwenhoek started looking at some kind of bizarre things with his linen microscope. And one of the things that he looked at first was peppercorns. You know, the little things that you grind up to make black pepper. And he was interested in, why is pepper hot? Why is it spicy? And he thought, well, maybe I can get some insight into why it's spicy by looking at it. Structure determines function. Okay. So he took some peppercorns and he kind of mashed them up and he put them in a little bit of water so he could get a little bit of droplet. And when he did that and looked at them, he saw little tiny microscopic things, and these are some of the von Leeuwenhoek drawings, which turned out to be bacteria. And as he looked at other types of water, like rainwater or water from a pond, he saw this whole new microbial world. And it blew people's mind. Okay? All these invisible little microorganisms swimming around, and people just were freaked out by it. He presented his results to the Royal Society of London, which was the preeminent scientific organization of the time. Isaac Newton founded the Royal Society. And it was a big deal. And it opened up this whole new idea of there's a whole new world out there that we can't see. And some people started to think, I wonder if there's a relationship between all of these tiny little invisible microorganisms and human disease. And that story about how that was all teased out is something we'll follow, and it's going to have some implications for some things we'll be doing in lab. So, tomorrow, be here at 9. Probably only take about a half an hour. I'm just going to get you started, and we're going to have to take out. You won't need your lab manual by tomorrow. Also, tomorrow's the time to pick where you want to sit for lab. Okay, so when you come in, plop down and where you want to do your lab. Okay, you guys failed me. You didn't remind. I got it. You didn't remind me.
Oh, I was gonna